Good evening, everyone. Whew. Welcome to the last of our 15th anniversary series. So many of you in this room have been here all year for the many different events we've done. I'm just curious, if you've been to another anniversary series event, raise your hand. Okay, that's just about everybody in the room. That is fantastic. That's why we did it. We wanted to bring people back to the campus, make sure you are connected with what we're doing, and hopefully be as excited about that as we are. So we're delighted to have you here. I want to say a special thank you to Mark and to Evolution, because you have sponsored this entire anniversary series, and we thank you for that, Mark. Absolutely. I also want to thank Penny Keene and the Keene Group. Penny's not here this evening, but she and Morgan Stanley and the Keene Group also were a sponsor tonight. And then Service First particularly sponsored tonight, and you guys got here just in time to hear me introduce you. So Delenn, May, thank you very much. We're so appreciative of you all making this event possible so that we could offer it at no charge for people to come. So without further ado, I'm going to turn it over to Dr. Sarah Sharman, who's going to get us started this evening.
Thanks, Elizabeth. I'm so pleased to join you all tonight and moderate another of the 15th anniversary series seminars. Tonight, we're going to have an impactful discussion about HudsonAlpha's research and the importance of genomics in our world today. Tonight, I'm joined by a familiar face to most of you, Dr. Rick Myers, HudsonAlpha's Chief Scientific Officer, President Emeritus, and M.A. Loya Chair in Genomics. Sitting next to Rick is his longtime friend and our honored guest, Dr. Eric Green. Dr. Green is the director of the National Human Genome Research Institute at the National Institutes of Health. You'll probably hear it referred to as NHGRI for the rest of the night, because that's just a mouthful. Dr. Green was appointed as the NHGRI director in 2009. A few years before that, both Rick and Eric played integral roles in the Human Genome Project, which we'll learn more about later tonight. In just a moment, Rick will give you a more detailed and personal introduction to Dr. Green. After that, Dr. Green will give a brief presentation about his work with NHGRI, and then I'll lead Rick and Eric in a discussion about discovery, innovation, and progress in genomics. Rick, you're up.
Okay. Thank you, Sarah, Elizabeth, everyone, for helping to arrange this, and thank you all for coming. I promised Eric that I wouldn't tell too many stories about him because he has more to tell about me, so I need to be a little careful there. We met in the late 1980s. I'm significantly older than Eric, so he was, I think you were still maybe a fellow, MD, PhD, with background in actually pathology. But we met because we were just starting to discuss the idea about having the Human Genome Project. People were, like my lab and many others, were looking for genes that cause diseases when they're mutated, and it was really slow. The cystic fibrosis gene was found back then. There were several others, and the idea came, the idea that we ought to just figure out where all the genes are in the human genome and then use that to build on other things. And you're going to hear more about that tonight. But I met Eric early in those days at a conference, and we quickly became friends, but also sort of collaborators and colleagues all along the way. So, and one of the stories I will tell that you'll hear about in the Genome Project, there are some similarities between us. He's a little bit taller, but also we both have a lot of energy. Eric actually channels his energy extremely well. He's the most organized human being on the planet, and nobody, Wendy, would accuse me of being that, although I'm not terrible. And so that showed up in the Genome Project. And one of the things, this was a new project. You'll hear more about it, but we didn't know what we were doing, frankly. We didn't know how we were going to do it. And a lot of Eric's sort of standards helped all of us, and this was ultimately 2,000 scientists or so, sort of keep our eyes on the prize. So I'm really glad. This is Eric's 4th visit here. He's been not only in his job as NIH and the director of this wonderful institute, but many other things having to do with training and things like that. So Eric, thank you so much for coming back again.
And by the way, I just want to point out that this photo was about 9 years after we first met, because when we first met was about 1988, just before the Genome Project started, and this is at about the halfway point. This photo was taken at a meeting at Cold Spring Harbor, New York, at about the halfway point of the Human Genome Project. So I was even younger looking when you first met.
Well, and we had more hair back then, too.
Yes, that too. Should go?
Okay.
All right.
So Eric's going to give us the story here, and then we'll watch a video afterwards.
Well, thank you, Rick. I'm delighted to be here. I jumped at the opportunity when Rick wanted to have me be part of this 15th anniversary, and I'm happy to be here tonight and tell you a little bit about the passion that Rick and I have in genomics, but in particular what we're really looking at now, as you can see from the title, of moving genomics to the point where it actually is going to positively influence how we practice medicine, something I'll refer to as genomic medicine. But to tell the story, I do want to just sort of briefly remind you a number of things that are really quite relevant. First of all, Rick and I were both attracted to this field of genomics, not because the field existed while we were getting our formal education in graduate school, or in my case, medical school, because genomics didn't exist as a field until 1987. The word genomics didn't exist until it was coined in 1987. And shown here is the first time the word genomics was put into the scientific or medical literature was in a brand new journal they called Genomics, because the word had just been coined. And never once in the formal education that Rick had, or I had, including graduate school, medical school, do we ever hear the word genomics once, because we had already graduated from professional schools when the word was invented. But both of us were incredibly attracted to this new field for a number of reasons. And the reason why the field was getting so hot was because of this audacious project that was being undertaken called the Human Genome Project. So I now direct one of the 27 institutes at NIH. You heard, we refer to it as NHGRI. It's the National Human Genome Research Institute. It is one of the 27 institutes, but we were created explicitly to lead the US's effort in the Human Genome Project. So we're still a relatively young institute at NIH. But Rick, and even before I got there, and similar to Rick's entry into the field, we both got involved in this incredible project called the Human Genome Project, which went from 1990 to 2003. And I'm sure we're going to talk about it more during the discussion. But this was, for both of us, career transforming. I don't think any of us went into our profession expecting that we were going to work on a project like this, but both of us have made a career by, at very early stages in our professional pursuits, got involved in this incredibly wonderful project, international in scope, very large, big, organized effort that ultimately was successful and achieved all of its goals. Now, as you will see, Rick and I have aged extremely well. You already saw this photo. And this was during the Genome Project. Unfortunately, we don't have any photos of when we first met in Tahoe. And the other thing I noticed, we don't have a whole lot of photos of us together, because either one of us is taking the photo or one of us is in the photo. So this is the only one I could find of us together in a photo. This is a good photo of Rick during the celebration when the Human Genome Project was completed, and there was a formal gala, if you will, to celebrate that, and he's with two of our good colleagues that participated in the Genome Project. And then I came to the opening of the HudsonAlpha, and there was a black tie event, as you can see. And so these are photos that I have of various events associated with, I guess, 15 years ago or so, when I was the first visit I had to this campus. So yes, Rick and I go back, but more importantly, we are aging extremely well. So this is a big year. Remember, the Genome Project ended in 2003, and we are now at the tail end of 2023. And all year long, we've been celebrating the 20th anniversary of the Human Genome Project. And we are not the only ones celebrating this or talking about it. It is sort of a really important odometer moment, thinking back on 20 years. We now have had laid out before us a complete sequence of the human genome. We ordered the 3 billion letters of the human DNA blueprint, And we've had it in front of us now for 20 years and pursuing the sort of what's next. And a lot has been written about it this year, and I'm going to briefly go through what I think has been the major highlights of the last 20 years. But I did want to use this quote because this isn't even a scientific publication. This is just the magazine The Economist. And they even commented this year recognizing that 20 years after the Human Genome Project, it's really revolutionized biology. And I just love this quote from it. They say, "No part of biology is untouched by the consequences of the Genome Project. Genomics has therefore come to form a framework for biology in a way that the periodic table forms one for chemistry. It touches everything." Now, that's a pretty impressive quote, and so I want to sort of briefly tell you about the last 20 years in particular, because it really takes us up to the present time to see really why an institute was created at the National Institutes of Health that was going to help lead the US's effort in the Human Genome Project. Because the rationale that Congress saw that prompted them to give money to the NIH to create our institute and to pursue a major contribution in the United States for the Human Genome Project was a recognition that if only we could empower physicians to have information about each individual's DNA blueprint, maybe it would improve the practice of medicine. I can tell you that everything I learned in medical school, because I graduated in 1987, I was taught how to treat the average patient, because that's the only way we had to do it, just to do based on averages. But none of us is average. Each of us is unique. And the way we are unique is our social existence and many aspects of our lives, but also a key part of this is each of us has a unique DNA blueprint that is slightly different from everybody else. And so the key rationale from the beginning of the Genome Project was we could potentially use genomics to improve how we practice medicine by changing it from a paradigm where patients are treated on average, where patients are treated more personalized based on their unique genome. So it wasn't terribly surprising that as soon as the Genome Project ended, the idea of juxtaposing the word genomic and medicine made a lot of sense. You would see that in the New York Times, where they talked about genomic medicine, and then scientific journals featured the concept of genomic medicine. The only problem with these publications is they made a lot of sense when you put two words together, but 20 years ago, it was a very blurry concept. We really didn't understand what it was really going to mean to encounter a patient and use information about their unique genome to tailor their medical care. And so in some ways, that became a second major journey for this field of genomics. So the field started in '87. Its first journey was the Genome Project, get a sequence of a human genome. We did that until 2003, and this was going to now be the starting line. We had the sequence of the human genome, and we now wanted to realize genomic medicine. And that's what we've been doing for the last 20 years. We didn't really know all the steps that were going to be required. Of course, we didn't know all the steps for the Genome Project, but we just figured it out as we went along. And every time we would accomplish a step, we would cross it off our list. We'd go check, 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 and eventually we got to the end of the project. And we also knew that the journey to genomic medicine was going to be a lot like the Human Genome Project, in that it wasn't just going to be done by one lab, not just Rick Myers could do it, not just I could do it. We were going to need hundreds and hundreds of scientists and thousands of people working on it, and not just from one discipline. It's going to have to be many, many different disciplines. And I can also tell you that we knew getting to genomic medicine was going to take longer than the Genome Project. In fact, we're only part of the way there even now. It's going to take decades. So it's more like a marathon where you have many people running shoulder to shoulder, many different disciplines, internationally helping to share a lot of information and inch our way closer to using genomic information about individual patients to improve their medical care. So what has happened in the last 20 years? What are the major steps towards really making genomics relevant in medicine? Well, if I had an hour, I would tell you about this in detail. I'm going to try to get this over in about 3 or 4 minutes, this slide. And so I'm going to condense 20 years of progress into about 2 minutes. But here we go. So the first thing we did, I already told you about. We sequenced the human genome for the very first time by the Human Genome Project, the starting line of the next pursuit, finding our way to genomic medicine. One of the first things we needed to do was we needed to make it cheaper to be able to read out an individual person's DNA sequence. because that first sequence of the human genome cost something like $1 billion. We needed this to be a clinical test. We needed it to be like a cost of a CT or an MRI. And so we set our goal, let's get it down to $1,000. And guess what? We're there. We've now reduced the cost of sequencing a human genome by over 1 million-fold. It is now less than $1,000 to sequence any one of your genomes. We can cross that off our list. Huge, huge accomplishment of the last 20 years. Oh, wow, if we have a cheap way to sequence genomes, we wanted to go from the one sequence we had of a human genome by the Human Genome Project to understanding what all of our genomes look like and increasingly understand the small, small differences that exist across each of our genomes. By the way, what do I mean by small, small differences? Compared to the person sitting next to you, either on your left or on your right or in front or in back, there's probably about 3 to 5 million letters of DNA that are different between you. You're still 99.9% identical to that person, but there's still 3 to 5 million individual differences. And we are getting, because we've gone out and used our new approaches for sequencing DNA, we've now sequenced millions of people's genomes, and most of the spelling differences in your genomes we already know about. We've already sequenced and found them in other people and have made those available on the internet in databases that allow us to study the relevance of those DNA changes. So we're now gone from 1 to over millions. But of course, just because you have a letter difference in your DNA, sometimes it's important and sometimes it's not. Sometimes it's consequential, sometimes it's not. We need to figure that out. In order to do that, you need to understand, well, how does the human genome actually work? How does it function, and how does spelling differences in it influence how it functions? And through work that has included some incredibly important work that has gone on here on this campus in projects like ENCODE, and I'm not going to tell you a lot of details, but this campus has contributed significantly to figuring out how the human genome actually functions. And while we have a long way to go, and this is something that Rick is an expert in, we have made great progress in 20 years. We still have lots and lots to learn about the genome. It's a complicated 3 billion letters, but wow, we know a lot more now than we knew 20 years ago. And as a result of it, we have now been able to significantly advance our understanding about how DNA changes influence our health, lead to disease. In some cases, it could lead to breaking one gene that causes a rare genetic disease. You heard about cystic fibrosis. You know about diseases like sickle cell anemia and Huntington's disease. Well, here's a number that I think is really impressive. Those are all rare diseases. When the Genome Project began, literally the day the Genome Project began, there were 61 rare diseases that we knew exactly what gene in the human genome had a mutation in it to make it not work. One of them was sickle cell disease, one of them was cystic fibrosis, and there were 59 others. Today, it's nearly 6,000 rare diseases we now know exactly what gene is mutated. We've gone from 61 to nearly 6,000 since the Genome Project began. Now, most diseases that you're more familiar with are not rare diseases. They're more complicated. And we are making great progress because it ends up being multiple genes. There's a lot of complexity, including a lot more influence of the social and physical environment. But we have made significant advances in unraveling the genomic basis of human disease, although again, we have a long way to go, but wow, we can look now in the rearview mirror and say where we were 20 years ago, see where we are now, and really recognize a tremendous amount of progress. But as a physician, what I'm actually the most excited about is the fact that there's one more highlight, these major steps, because over the last 7 or 8 years, we have begun to see vivid examples of genomic medicine now emerging. When I mean genomic medicine, I don't mean just understanding when a DNA change leads to a disease. I mean actually changing how you practice medicine by having genomic information about a patient and actually using that for clinical management. And the earliest examples have now emerged. And the reason they have emerged is as follows. If you have a patient, and under a certain circumstance, you think it's important to understand DNA of that patient, you can read out their genome. It's pretty routine, it's not expensive, it doesn't take very long. And what do we do with that information? Well, we simply are now gotten better and better at sort of recognizing that there are differences in our DNA, these letter differences, one position, different people will have different letters. And what we do is we simply compare a patient's genome sequence, shown on the right in purple, and we just compare it to a very high quality, sort of certified, we refer to it as a reference sequence. It's just sort of like the gold standard. And then you just make a list of all the spelling differences. We call them genomic variants. They're just spelling differences. And you use that information and then look to see if it influences the function of the genome and what that might mean, whether it makes a gene not work, et cetera, et cetera. And that's the paradigm. It's a diagnostic test. What this has happened in the last 7 years, it has brought genomic medicine into focus for the very first time. And I will tell you that 20 years ago, when the Genome Project ended, many people did not think genomic medicine would have any real-world applications for probably 3 or 4 decades. And we have them now already 20 years later. What are my highlights here? And again, I don't have time to tell you in detail about these examples, but what I would say is that none of us expected any of these things to happen as quickly as they have happened. Probably where you will see genomic medicine have its highest impact in the long run, and you already see it for some situations, is in the arena of cancer. Cancer is a disease of the genome. The reason you get a tumor, the reason you get cancer is those cells have genomes that have had their DNA changed so much that those cells grow out of control. And guess what? You can read out the DNA of those tumors and learn a lot about them, including how to better manage that patient with cancer. It is absolutely standard of care for a series of cancers already. That list will grow. I believe in the long run, oncology is going to be the place where genomics is going to have the greatest impact. Another area, including stuff that goes on right on this campus, is in the arena of rare diseases. Increasingly, when you have a patient and you think they have a genetic disease and you don't know what it is, you sequence their genome. It's relatively inexpensive, less than $1,000. You can get the results back, and in a great fraction of the time, you will figure out what is wrong with that patient. In some cases, it's just a routine clinical involvement. There's not an urgency. But in other cases, there is an urgency. And I will tell you that in neonatal intensive care units in the United States and in other places around the world, there are almost always children where the neonatologist has no idea what is wrong with that child. In fact, could predict the child might not last another few days. And there are now routines that allow that baby's genome to be read out in less than 20, and interpreted in less than 24 hours. And it is saving lives, and it is absolutely miraculous that it could be done that quick. And this is becoming routine. That's a baby that's born. What about an unborn child? Well, prenatal genetic testing has been going on for years. My wife, or both of our kids, underwent amniocentesis in each case, where a needle was put through her belly and a little bit of amniotic fluid was removed to get DNA of our unborn children, and we made sure the chromosomes had all normal numbers, the babies were healthy, and those were prenatal genetic testing, which thousands and thousands of women have gotten for years through an invasive procedure like amniocentesis. It's not done that way anymore. Turns out that our new methods for sequencing DNA are so exquisitely sensitive that all you need is a blood draw from the pregnant individual. And so the pregnant individual, they get blood draws all the time. You take one extra tube of blood, you send it off, and you can detect the small amount of fetal DNA that naturally gets shed into the maternal bloodstream, and therefore it can be analyzed in a fashion that gives you the same information that you would otherwise get through more invasive procedures. This is actually the #1 genomic medicine test worldwide. About 7 million pregnant individuals will get what's called non-invasive prenatal genetic testing done this year. Another area to watch for is a big word, pharmacogenomics. It's just pharmacology and genomics. Why is it that so many medications don't work? It's not because the pharmacy is incompetent. It's not because the physician is mean. It's just that there's not a very good way to match the right medication at the right dose for the right person. There's like 10 different hypertensive medications, and it's a very average thing. You try one. If that doesn't work, you try another. That's so imprecise. We're changing this because we're learning that the reason people respond differently to medications has to do, in a large fraction of cases, to differences in our DNA that influence how we metabolize drugs. And certain drugs are better matches to certain people based on these DNA signatures. So increasingly, what you expect over the next 10 or 20 years is They may want to get a genetic test on you before they give you a prescription to make sure they're giving you the best asthma medication, the best hypertension medication, the best antidepressant, et cetera, et cetera. That is pharmacogenomics. It's here and now for a handful of drugs. Expect that list to grow. And then the last area to look for, this is clinical research. It's not clinical care. My first 4 examples are real. My 5th one is on the horizon, is predicting more common diseases like diabetes or sudden cardiac death or different kinds of epilepsy by looking at people's DNA and making predictions whether they're at high risk or not. I'm going to put a question mark by these because this is an area of intense research going on at my institute and others at NIH, because it's not clear yet it's ready for clinical implementation, but it's something that you might hear about. You'll hear phrases about polygenic risk scores. I can explain that later if you're interested. But it is, I think, what might be the next thing to look for in genomic medicine. So this is why I'm profoundly excited and profoundly proud at what's happened in genomics when it comes to applications towards human health. Well, what about the next 20 years? Well, we are constantly landscape surveying, and we're constantly trying to push the frontiers of these areas that I've talked about and even more, and we regularly publish strategic visions. Our most recent one we published in the middle of the pandemic in October of 2020 that talks about our new strategic vision for pushing the envelope of applying genomics to improve human health. And later on in the discussion, we talk about a lot of scientific things in this, and then at the very end of this paper, we talk about a series of 10 bold predictions of what we think might happen over the next 10 years. And you're going to hear about that a little bit later in the discussion, so I won't say any more about it. So that was going to be the end of my talk. But then I got off the airplane last night, and I started to walk through the Huntsville airport, and I said, oh my God, I'm in Huntsville. Oh my gosh, this is like the home of incredible aviation advances, and they're so tied to so many things going on with space exploration. And I always like this metaphor. I should have a slide for it. So last night, I created a new slide just for this talk, and it sort of embraces this idea of how proud Rick and I are of what the Human Genome Project was all about and what this field was about, because we can totally identify with the idea of the moonshot. Because in many ways, the moonshot was incredible, and the Human Genome Project is biology's equivalent of the moonshot. There is one difference. I mean, so the moonshot, I don't need to tell an audience like this all about it. I was a youngster at the time. It was incredible. And typical of space exploration, it involved a very large managed project. It was estimated about $25 billion to put the first person on the moon. And its signature goal was incredibly successful. You put a person on the moon. And if my Google searching is right, that's been repeated about 11 times. So the moonshot accomplished its goals and has done it 11 more times since it accomplished its goals. I'm particularly proud of the Human Genome Project, which I think is biology's equivalent of the moonshot. It was biology's first large managed project. Space exploration, you're good at managed projects. Biologists, we had to figure it out for the first time. A lot cheaper, $3 billion. But what I'm particularly impressed by is that our signature goal is sequence a human genome. We've now repeated it 1 million times, actually far more than 1 million times. And so that's really pretty incredible. And of course, that's been accomplished because we were able to reduce the cost of sequencing a human genome by more than 1 millionfold over the last 20 years. So in any case, I think these 2 things represent 2 of the most impressive accomplishments in the history of humankind. And I know I'm biased, and I'm sure all of you are as well, but I am immensely proud to be able to make a slide like this and say, here we are. What Rick and I and our colleagues have done, I think is sort of the moral equivalent in many ways of what the moonshot was all about for space exploration. So with that, I will stop, and we'll have a discussion.
Thanks so much for taking us on that journey through the history of genomics and genomic medicine. I'm so impressed you fit that into 15 minutes. I can't wait to dig deeper into some of the topics you were talking about. But first, I think I and a lot of people in the audience would like you both to take us on a trip down memory lane. So I think it's so fascinating you both worked on the Human Genome Project. As a science person, I'm really nerding out sitting here on stage with you guys. So what was it like behind the scenes in the early days of the Human Genome Project? Do you have any favorite stories or memories you can and want to share?
So, I have lots I could share. I really want to emphasize a point. I'll pick up on a thread. Rick said it briefly. I think there's sometimes an assumption, and maybe because our PowerPoint presentations are slick and we just sort of talk like, oh yeah, we made it happen, that we had any idea what we were doing when this thing started. I can't emphasize enough that the Genome Project started. We had a goal. Get us, there's a series of goals. The ultimate signature goal, get the sequence of the 3 billion letters of the human genome, and we had absolutely no idea how we were going to get there. I mean, it really was one of these things where you just had to take one step at a time, and it was terrifying at times, and there were so many speed bumps along the way. And Rick and I can both echo, because we still hear it in our heads, people who had advised each of us that we were probably committing career suicide doing this. Some people had the attitude at the time that the Genome Project was mundane, it was going to be boring, that it was really more appropriate for prisoners to be doing it than scientists. I can certainly tell you that I was being told I was going to throw away my medical career by getting involved in this. And Rick and I ignored that headwind and said, no, this just feels right. We should be doing this. But when we would hit speed bumps, including threats of the private sector and things that just weren't working, it was terrifying at times. I mean, it was scary because you were not only worried that you were going to be embarrassed, you were worried that you were going to be throwing away your career. So it was not always smooth sailing, but I don't think it was, it always felt like you were part of history.
So one of the neat things that happened is, I mean, first of all, this was a major team effort. It involved countries all over the world. The US and England really led the way, but many groups in Asia and in Europe were participants. And I think the reason it worked, clearly, the technology that we had in 1990 when we started, it would have taken us another 100 years maybe to use that. So it had to improve. It really had to improve. And it wasn't just getting the cost down, it was getting the accuracy up, making it where it was incredibly labor intensive. But as the project developed, and especially when sequencing really in earnest started in the late 90s, we were mapping and getting all the technologies ready. When sequencing started, it was much better, but it still took more than $1 billion to get all the way through to the first one. And it's just remarkable. Not only did we have millions of human genomes, all along the way groups were then starting to sequence the genomes of other organisms. And our group here, now led by Jeremy Schmutz and Jane Grimwood, who are two faculty, were at our Genome Center at Stanford and really played a major role in making that happen for our group. Then switched when we finished to other organisms, and a lot of you know this, we are major players in the world for genomics of plants and really having to do with sustainable agriculture and many things well beyond that. That's one of the really neat things about the Genome Project. It was technology that had zillions of applications, and we're helping to be part of that as well. But I agree with Eric. It was scary because we really didn't know. I mean, there was that career suicide worry. It is true that when we first started, when it was first proposed and we first started, there were a substantial number of prominent biologists who were dead against it. And it was really, and I'm not one of those I told you so people, because it wasn't my vision. I got to be part of it, but it was really wonderful to watch essentially every single one of them come around to realize, oh my gosh, this is incredibly valuable. That's one of the fun things about the Genome Project.
You wanted a funny story? I can think of a lot of funny stories, but usually people like to hear this one. I don't know if Rick was at this meeting or not, but inevitably, when you talk about the Genome Project, one of the most common questions that Rick and I certainly get is, well, whose genome did you sequence in the Human Genome Project? Well, it turns out it wasn't one person, it was a whole series of people, and it's sort of almost, think of it like a quilt, a patchwork of different people, just sort of little parts of the genome are just from different people, and it's fine. Since we're all 99.9% identical, it doesn't matter for that first sequence of the genome. But it was such an obvious thing along the way that, you know, we're going to have to use somebody's DNA when we started sequencing the genome. And it got to a point where we had made enough progress, it's probably about 1995, that there needed to be some very clear indication of what DNA we were going to use when we were going to start to sequence the human genome. So there was a meeting, I think it was in Bethesda, I don't know if you were at it, where that was really being discussed. All right, we're going to start sequencing the human genome. We got to get organized. We got to get all the DNA libraries made, everything ready to go. And I remember there was a whole session about, well, whose genome are we going to sequence? And there was a lot of discussion about, well, who should it be? How are we going to get the consent, blah, blah, blah. And I remember, I don't even remember who it was, but some scientists, because it was all a bunch of nerdy people like Rick and I talking in this room, you know, and we're all just geeking out about all this. And somebody raised their hand, they said, well, I don't know whose genome we're going to sequence, but whichever person gets picked, they have to be normal. It's like, and how do you define normal? So, and trust me, there's not a person in this audience who's normal. Every one of us is a mutant. We all have differences. So it's just, and the room just went quiet. And I just, you know, and this was a very smart person, but, you know, just goes to show.
So let me say one thing about that Eric alluded to, but when we do these sequences, from day one, the Genome Project, the U.S. Genome Project, had an effort called ELSI, but it was really, which stands for Ethical, Legal,
Social
Implications. And a big part of this that already existed, and a lot of you have heard us talk about this, was genetic privacy. That's really one of the biggest issues. And so we have, when people get their genome sequenced, unless they choose to make it available to make everybody know that it was theirs, it's completely private. And that's very important, although I think we've dealt with a lot of the issues that would arise. We helped to start, a bunch of us, including Eric, helped to pass a law called GINA, Genetic Information Non-Discrimination Act, that on our starting day, Jim Hudson, you're in the audience, I think, we were in a room in our building there on the day that the Senate was voting to pass this law. It was the final step. Francis Collins was in the room. Eric was with us as well. And 99 to blank, because one of them couldn't vote. And so really important things. And there are many more aspects to that we continue to make important. One of our faculty members is a bioethicist. We do a fair amount of that stuff. The genetics counselors, who are, many of them are in the room here, make sure that we maintain that for privacy reasons.
By the way, when was the last time you got 99 senators to agree on anything? That was a different era.
Yeah, different world then. 2008.
I think we can all agree that the Human Genome Project triggered unprecedented momentum for new technology and innovation in the genetic field. Dr. Green showed us a lot of good information earlier, and he mentioned a video that NHGRI put out, so I would like you all to watch it now, and then we'll discuss it afterwards.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5. Student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6. Genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7. It will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8. A person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9. Advances from human genomics will benefit all of society and not just a few. Prediction 10. Genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
Such an inspiring video. For the rest of our time, I just wanted to focus on a couple of those predictions that I found interesting, and I'm hoping members of the audience also found interesting. So, Rick and Dr. Green, why is it important that sequencing and analyzing a genome becomes commonplace for every research lab?
Yeah, I mean, it picks up on a theme that Rick implied in some of his earlier comments. I think so much about genomics is about lowering barriers. I mean, what used to be years and years of work to study one gene, the reason we did the Genome Project was to make it just something you look up on the computer. I mean, that's what sequencing the genome allowed you to do. The reason we wanted to reduce the cost of sequencing a millionfold was to not have it just be done by a big consortium, but have it be done anywhere. And that prediction was put in as, yeah, isn't it incredible that we can sequence human genomes at Harvard, at Stanford, at HudsonAlpha, at NIH. But we want to continue to lower the barrier so that less resourced institutions, less resourced parts of the world, anybody doing any research, it's just maybe, and including in student laboratories in colleges, maybe even in high schools, can sequence genomes. And by doing that, it empowers more and more people to use the power of genomics to, one, get excited about science, but also to do scientific discovery. So it's all about lowering barriers.
One of the neat things is that it's one thing to produce the sequence or to generate the sequence of genome, but what does it mean? And this is a place where it really has and can spread around the world, everywhere, which with students, even high school students, even sometimes younger. And I don't know if Alex Harkis is here, but he's doing this program with students for a project in plants. But the idea that you have to analyze it. And the truth is, I would say that probably more than half, certainly more than half of the effort now is figuring out what it means. And that even with new tools, and those tools are getting better, but even with new tools, it still takes effort to do that. And it doesn't always take enormous computer power to do it. So it's been really fun to see that being spread around the world, and also in sort of inspiring younger folks to come in and help us do this.
Yeah, I mean, to do state-of-the-art genomics now, you need a laptop computer. I mean, you don't even necessarily need a laboratory. And what we're seeing happening all over the world, but especially here in the United States, lots of programs to try to get lots of people, not just at high-resource research institutions, but all over doing genomics. If nothing else, they could become data scientists and analyze data. By the way, when the Genome Project ended 20 years ago, we tried to attract the brightest minds in computer science to get into genomics. I remember having workshops where we invited all the smart computer scientists at MIT and Harvard and Stanford and all the places like that. They were not particularly impressed because we said we have 3 billion letters. They said 3 billion, that's not that much. Now, with a million genomes sequenced and tons of other data, we are very much the envy. We have all the brightest minds working on genomics, bringing AI and ML and all the fancy stuff, because now we are at the big data table, and that makes it very exciting.
And biology is complex, so it's not just this linear array of 3 billion bases. And so engineering helps a whole lot, both in the wet lab and in the dry lab stuff, but it's not a simple engineering problem either because of the complexity of life. It's hard to predict what's going to happen based on even all the information we have. We're getting better and better and better at it, though.
So we have millions of genomes now, and that is a lot of information, and it's really helping scientists kind of understand the function of our genes and our genome, especially for human health and disease. Are there any recent or ongoing studies either of you are exceptionally excited about right now?
So there's lots of them. So by the way, there's the side I want to point out, and is that Rick and I, among our many similarities, we are exquisitely enthusiastic and incredibly proud of what's happened. But on the other hand, I think we're both realists. That I kept saying in my remarks that we've just made incredible progress, but we have so much more to go. I'll give you an example. And this project that we're funding, we're very excited about, is. And maybe you should then talk about the next kind of things beyond the genes, is that, here's what we know. 20 years after we have the sequence laid out in front of us, we have a really pretty good inventory of all the genes in the human genome, the genes that directly code for proteins. There's about 20,000 of them. That's the good news. The bad news, or it's not bad news, it's just that the realism, we know only what a small fraction of them actually do. For the great majority of those 20,000 genes, we don't actually understand what they're doing. So that's, and so this is a huge deal. And so we have a project, it's coming directly out of our strategic vision called Morphic, the name doesn't matter, where we're going to really try to figure out what can we do to figure out, at least at a fundamental level, what each of those 20,000 genes could do. But that's the genes. And that's only part of our complexity, where the guy sitting to my left is one of the world's experts in how the genes, the choreography that makes all those genes operate. So maybe say some things about the choreography of gene regulation, which you've spent your whole career thinking about.
Yeah, that's true. We call it regulation of gene expression. What that really means is how do genes get turned on and off? We have 22,000 genes and a bunch of other regulatory elements millions of regulatory elements that control them. And that's where a huge amount of biology and therefore a huge amount of health and medicine come from. And that's what, and if you ask me what I'm looking forward to, that is more than incrementally, but it's getting better and better. And I think we're going to have an understanding of that, and partly because computation and things like that are helping, as well as more and more experimental approaches. I think all of us want to see this be practical. And I have to say, when we started the Genome Project, it wasn't that I wasn't skeptical. I dove in full blast. I was incredibly excited. But I honestly had no idea we would be using this already. Even a few years into the project, we were finding genes that cause disease and actually being able, a big part of this is diagnosis and being able, if you know something is going to happen early, you can actually intervene a whole lot better, especially if you can catch it before symptoms even start. That's true not just for cancer, but for all sorts of diseases. That's where we're heading, and I think we're going to see a whole lot more of that now in the next 20 years, next 5 years.
So I really loved the projection that students will do large-scale genomics projects in school, and we're seeing a little bit of it now at HudsonAlpha, at least the high school level and up. But I assume in order to get kids interested in STEM, we also need to reach their parents and teachers and other influential people in their lives. So how important is public outreach and education in fostering a more genomically literate community and giving children a fair opportunity to get into STEM in the first place?
So I have an answer to that. We need to clone Neil Lamb and his offspring. I'm serious. When we, really 2 years before we started, we met Neil and hired him even before we opened the doors, a couple years before, to build this education outreach program. Kelly East runs it now that Neil is president. I am so proud of this because I don't think it exists anywhere not like this, and it needs to be everywhere, things like this. And part of that, it's not just the next generation, it's, I mean, I think we have more adults coming to some of our programs than we do children, or kids. I mean, it's some of both. And part of that is the, first of all, to be honest, brokers of the information so that you know the pluses and minuses, or at least some of the things to pay attention to, but also to inspire. I wasn't just kissing up to Neil then, although I was doing that.
No, he's your boss. So I completely agree with and echo everything that Rick said. And the only other dimension of this that I tend to stress as a physician scientist is the honest, true belief that genomics is going to become mainstream in medicine. And all of us, every person in this room, we're all going to be patients, or we already are patients, or we have friends and relatives who are patients. And as more and more genomics is spoken as part of routine medicine at all life stages, it's really going to be important and empowering to make sure everybody has a minimal amount of genomic literacy, because it's going to be part of the language of healthcare decisions that you're going to need to be a part of. So that's just as a very practical, everyday life, it's just so important.
So let me just add one thing to that. Part of that is not making the physicians and the healthcare providers have to be absolute genomic experts. And so a lot of the programs and ways, and in fact, I think this has been one wonderful thing about our education outreach program and our genetics counselors, is to figure out how to make that digestible and quick in the sense that you don't have to spend hours explaining to everyone about every last detail of it. And it turns out, I mean, it's getting, Neil, wouldn't you say, I think it's getting way better and better because, partly because we know a whole lot, but then also we have very talented folks figuring out how to communicate complicated things.
So in the video, you all saw that it projected that advances in human genomics will benefit all of society and not just a few. I thought that was such a powerful statement. So how is the field helping to make sure that genomics is accessible to all?
I mean, this is something we are just incredibly passionate about. You know, there's just been such an unfortunate history that, as we've seen over and over again in humanity, whereas technological advances found their way into medicine, those benefited people disproportionately. And genomics wants to do this differently. Now, it's going to be hard because the healthcare ecosystem is a very complicated ecosystem and it varies from country to country, et cetera, et cetera. But we really are doing a lot of things and laying a lot of groundwork. And again, it's all about lowering the barrier. It's all about making things accessible so that it's not just some that benefit from genomic advances in medicine, but that everybody can benefit. And unfortunately, we're going to have to apply genomics into a very complicated and imperfect healthcare system in the United States where there are naturally lots of disparities. But I just hope we don't exacerbate them, and we're doing everything we can to prevent that, and we're doing a lot to try to reduce them. But part of it, you know, is also to just keep getting people excited about this, getting people engaged in this, getting people literate in this, and thinking about not just how to practice genomic medicine at a wealthy academic medical center, but thinking about all parts of the United States and all different ecosystems where healthcare is delivered.
One of the goals from the beginning was that, and people were concerned because it was $1 billion to sequence the first genome, and it's less than 1,000 now, and it's going to be even less. It's not just that; it's all the other things that you need to do. is because of the benefits of actually catching something early, knowing this, once you have your genome sequence, it only needs to be done once, protected database, you share that information with your healthcare providers. It has enormous potential for making things, reducing our healthcare costs. Use the pharmacogenetics example here. And I experienced this myself. I've had loved ones who've experienced it, and many of you know this as well. You might have something, you're prescribed a drug, the physicians may be just fantastic at this, but it doesn't work for you because it varies because of the genetic differences. What if you knew that at the very beginning? I don't know if this is still the case, but it used to be, if you have asthma, you try 7 drugs before you find something that actually works for you. I mean, it's things like that. Imagine being able to, every time somebody has cancer, to be able to catch it early and give the right treatment for it. And we are heading, I mean, it's not the distant future. Some of that's already happening here. So I mean, while it's not only about money, what if we could do a whole lot more with less cost for our healthcare system? And I think that's one of the big goals.
So I think a theme this evening is just how quickly that the field of genomics has been moving, and it was just almost unpredicted. And it's going to keep moving this quickly, as we've seen. So Rick, can you just briefly mention maybe what role HudsonAlpha has in keeping the discovery moving at this quick pace and really just contributing to the field of genomics growing?
Yeah, I mean, there are lots of things I can say about it. We came here because we just love this vision that Jim Hudson and Lonnie McMillan had now a little over 15 years ago. And part of that was, while we had freedom to do a variety of things, we had one mantra, which was genomics, genetics and genomics. They're very similar to each other. And so some of that's been technology development. Some of it has just been a lot of elbow grease and doing it really well. And I'm bragging on others here, not myself at all. And that's made a huge difference. I and several of our labs are very interested in how genes get turned on and off. And as you know, when you work in I've been working in that field since I was in graduate school in like the 1920s, or a long time ago anyway, that we love to say that it is a huge amount of biology, and it ties together with what you were born with, what you're exposed to, the environment, like Eric was saying. And I see that as HudsonAlpha, as small as we are, make big contributions in that. And I also want to remind you that that's in human health and disease and in sustainable agriculture / environment, all sorts of environmental things that we're experts. And those sound like very different fields, and they kind of are, but we overlap an enormous amount in that.
Thanks, Rick. So I could probably sit up here and talk to you two all night, but I think we'll probably wrap up and let our audience get back home. But I have one remaining question for both of you, just so we can end on a high note. What makes you hopeful for the future of genomics research? I know that's a big question, but.
No, I'm happy to take the first pass at it. In doing so, I want to sort of bring in one other point that I wanted to make, and now I can make it. You know, one of the interesting things about genomics is that almost everybody, especially when the field was created, there were which was at the beginning of the Genome Project essentially, there was nobody trained in genomics because the field didn't exist. Everybody to the field at the time, we were immigrants from other parts of biomedicine. Rick and I came from very different places. Rick came from studying gene regulation. And I came from basically being trained as a physician scientist. My PhD had nothing to do with DNA, but I went into clinical pathology, and I was very interested in the idea of using DNA diagnostics somehow in some vague way to eventually find its way into medical care, having no idea really what that was going to really look like. And so a lot of our anecdotal stories sort of comes from our worldview of where we came from, what attracted us to this field. And what makes me optimistic now is when I got into the field, it was in a very vague way believing that someday, at some future generation, they would actually be, if the Genome Project was successful, we would be able to change how we practice medicine, by looking at patients' DNA in some very analytical and thoughtful diagnostic way. I never thought it would happen in my lifetime. I wasn't even sure it would be in my children's lifetime, not that I had children at the time. But at every turn, I never thought we'd get, so, you know, I really wasn't sure the Genome Project was going to work. It was going to take 15 years. No, it only took 13, and we did it. Then we said we've got to make genome sequencing much cheaper. We're going to get it down to $1,000. I thought, it'd take 20, 30 years. Oh my gosh, it didn't take nearly that long. We did it. 14 years ago, almost to the day, I became the director of the Institute, and I said, we're going to start doing genomic medicine. That's the goal. And people said, you're crazy. We're not ready for genomics in medicine. It's going to take decades. And here, 14 years later, I show you all these examples. If you would have told me when I first got involved in genomics that I would still be alive, let alone still be working, let alone leading the Institute, was going to see a neonate get its genome sequenced in less than 24 hours, figure out what's wrong with them, change their clinical management, and save their life, I would have thought you were deranged. I couldn't believe, I mean, I would have never believed that that would be possible in my lifetime. And it's not only on my lifetime, it's happening on my watch. So the reason I'm optimistic is I've been consistently wrong. At every turn, when I would, in disbelief that something could happen so quickly, it's happened much quicker than I ever would have anticipated. And so how could I not be optimistic? And while those 10 bold predictions are audacious and over the top, and they're not all going to happen by 2030, I am convinced that by 2030, a couple of those predictions are going to be far closer to reality than I ever thought when we wrote them in 2020. And it's because it's this repeated pattern of me being wrong.
So I'll talk about my optimism in a minute, but he just triggered an anecdote. I remember we planned to start in 1990 and finish 15 years later. And we had lots of meetings with all the genome scientists getting together to work and hash it out and everything all along the way. And I can't remember what year it was, but I got up from one of those meetings to go to the men's room, and I come back, and it was now 13 years rather than 15 years. Yeah. So, and we did it, as he said. So my optimism is similar to what Eric's saying in that I was a little too conservative when we started. I thought, yes, maybe it'll happen, you know, when we're really, really old, and it started happening very quickly. But what I'm really excited about, I think there's no question that, and I don't want to put a year on it, but we're going to be 10 times better and faster and cheaper of all the things we just talked about, maybe within 5 years, and maybe more than 10 times better. So that means it'll reach every corner of the globe, not just the United States, but everywhere, which is really, really important. That makes a huge difference for so many things in our health and our planet's health.
Thank you both so much for being here tonight. I think that was the perfect wrap-up to the 15th anniversary seminar series. Before we go, I'm going to invite Danny Wyndham to the stage for some final comments.
I feel like if my mom could see me on stage with Dr. Sarah Sharman, Dr. Rick Myers, and Dr. Eric Green, she would say, "Son, what are you doing on that stage?" Well, I'm just here to close out the program. Dr. Green, thank you for taking time out of your busy schedule to be here and to condense 20 years of genomic progress and tie it into what goes on here in Huntsville, Alabama, into 10 minutes. That was fantastic. Thank you, Dr. Sharman, Dr. Myers, Dr. Green, for what was an engaging, enjoyable, and educational panel discussion. Tonight's event is the final event of what has been a series of events to commemorate HudsonAlpha's 15th anniversary. These events have been very popular, resulting in over 3,500 registrations across this year. Thank you for being here tonight and for those of you who have been at more than one of these events. I'm excited about the role genomics is playing in allowing us to understand how diseases are genetically predispositioned, how we treat them, how that results in personalized medicine based on our DNA. I'm just as excited about the role genetics and genomics might play for helping us mitigate and even prevent diseases in the future, helping us to increase our health spans. If these topics resonate with you, I'd encourage you to consider supporting the work going on here at HudsonAlpha's campus through a charitable donation to our Innovation Fund. We have designated the Innovation Fund for our fundraising in 2023 because it is the vehicle that we use to spur on innovation across the campus. Whether it is seeding a new idea, helping scale promising ideas, purchasing new innovative equipment, or hiring new innovative people, it's those dollars that are allowing us to do things that are not possible through our daily, ordinary operating budget. And we have used these dollars that you have contributed this year to fund some projects that have created short-term, immediate, positive results. If you do support the work going on here, we will commit to you to be good stewards of anything that you give. As our 15th anniversary series draws to a close, we're already planning events for 2024. So we'll announce the schedule for those early next year. Keep an eye out for that announcement, and we hope to see you in some of those events too. In closing, I'd like to thank you for all that you do to support the work and promote the work that goes on across the campus here. On behalf of the entire HudsonAlpha team, we'd like to wish you a safe and joyous holiday season, and we hope to see you back here again soon. Many of our panelists and many of our staff will stick around, but with this, we are adjourned. Thank you and have a good evening.